What if I told you that a diagnosis of insulin resistance, like that terrifying moment when you are sitting in a doctor's office being told you have prediabetes or type 2 diabetes,
Mm.
what if that isn't actually a sign that your body is failing you?
Right. I mean, it completely changes the entire paradigm.
Yeah, exactly. Because the standard narrative, the one that says your metabolism is just broken and your cells are malfunctioning, it is fundamentally misunderstanding the actual physics of what is happening inside you.
It really is. We've spent, gosh, decades treating metabolic issues as if the body is just this machine with a broken part. But when you look at the actual cellular machinery, the reality is far more intricate, and honestly, it's far more protective.
And that is exactly our mission for this deep dive today.
Right.
If you are navigating a new diagnosis or even if you are just aggressively trying to optimize your blood sugar and get some mental clarity back, we are completely throwing out the generic doctor's office pamphlets today.
Oh, absolutely. The generic advice just doesn't cut it anymore.
No, it doesn't. We have a massive stack of sources to synthesize, and the data is honestly staggering. We are pulling from a landmark Frontiers meta-analysis on the structural architecture of resistant starches.
Which is a brilliant paper, by the way.
It really is. And we also have papers diving into quantum bioenergetics, research on ketogenic neurology, deep literature on the gut incretin pathways, and even studies on vagus nerve stimulation.
So the goal here is to construct a really comprehensive, multidimensional roadmap for managing your glucose. But to do that, we have to dissect this topic from four distinct
Oh,
levels.
the four levels.
Yeah, we're looking at quantum physics, which handles the raw energy in your cells, then biology, which governs the ecosystem in your gut, anatomy, looking at your nervous system's stress wiring, and finally psychology, mapping how your blood sugar physically alters your brain chemistry and your daily mood.
It's so interconnected, and I promise you, by the time we finish unpacking all this, you're going to view your metabolism not as this simple, calorie-counting furnace, but as an incredibly advanced quantum engine and a delicate chemical ecosystem.
It completely reframes how you look at the food on your plate.
It really does.
Yeah.
So let's just zoom all the way in, past the organs, past the tissue, straight down to the subatomic level. Let's talk about the quantum engine.
Okay, to truly understand blood sugar, we have to look at the mitochondria. Most of us probably remember from high school biology that mitochondria are the powerhouses of the cell.
Right. The classic textbook phrase.
Exactly. But our sources on quantum bioenergetics detail the literal mechanics of how that power is actually generated. Inside every mitochondrion, there is this folded inner membrane containing something called the electron transport chain, or the ETC.
The electron transport chain, I mean, it sounds very industrial.
Because it functions exactly like an industrial power grid. When you eat a meal, whether it's an apple or a piece of steak, your digestive system breaks that food down. But the ultimate goal isn't just to make smaller pieces of food.
Right, it goes deeper than that.
Much deeper. The goal is to strip those molecules down to their basic electrons. Your body delivers those electrons to the mitochondria, where they are fed directly into the ETC.
So the electrons are the raw fuel.
Yes, exactly. The ETC is a series of 4 protein complexes. The electrons are passed down this chain from one complex to the next, and as they move, their energy is used to pump protons across the mitochondrial membrane.
Okay.
This creates a massive pressure gradient. And finally, that pressure is released through a molecular motor called ATP synthase, which literally spins, like a microscopic turbine, to generate ATP, which is the chemical currency of cellular energy.
Wow. Okay, let me make sure I'm picturing this correctly for everyone. Let's use a hydroelectric dam as an analogy.
I love that.
Yeah.
Go for it.
So the blood sugar circulating in my body is the water reservoir. Insulin is the signal that opens the floodgates, letting the water rush into the dam.
Right.
And those spinning ATP synthase motors inside my mitochondria are the physical turbines actually generating the electricity for my microscopic cellular houses.
That is a highly accurate way to visualize the mechanics. The lock and key mechanism of insulin is literally designed to open those cellular gates so the glucose can get inside and provide the electrons for the turbines.
Okay, so if I am developing prediabetes, what is actually going wrong with the dam? Because usually we are told insulin resistance means the locks are jammed or maybe the pipes are clogged with sugar.
Yeah, and that plumbing analogy just falls apart when you look at the quantum level.
Yeah.
Think about your modern lifestyle or the standard American diet. If you are constantly consuming highly processed, rapidly digestible carbohydrates, you are keeping your blood glucose chronically high.
Because it's absorbing so fast.
Exactly. Your pancreas responds by constantly pumping out insulin to open the floodgates. You are shoving more and more glucose into the cell, which means you are stripping more and more electrons and violently forcing them into that electron transport chain.
Oh, I see. I'm flooding the dam.
You are pushing electrons into the chain way faster than the turbines can actually spin, so the system backs up. Specifically, at complex I and complex 3 of the chain, the electrons get bottlenecked.
And what happens to them?
Well, because they are highly charged subatomic particles, they don't just sit there politely in a queue, they leak.
Wait, they leak out of the power grid. Where do they go?
They leak out of the chain and directly interact with oxygen and nitrogen molecules inside the cell. And when a stray electron binds to an oxygen molecule, it creates a superoxide radical. This is the genesis of what the literature calls reactive oxygen and nitrogen species, or RNs.
So it's basically toxic cellular exhaust.
Extremely toxic. These RONs are highly unstable. They will violently rip electrons away from anything nearby just to stabilize themselves. They rip apart the mitochondrial membrane, they damage the proteins, and they even mutate the mitochondrial DNA.
That's
It
terrible.
creates a state of massive oxidative stress. I mean, if this continues, the cell will literally destroy itself from the inside out.
Wait, if the cell is on the verge of exploding from this toxic electron leakage, what does it do? I mean, it can't just sit there and die.
No, it acts in self-preservation. To stop the catastrophic damage to the power grid, the cell makes a calculated biological decision. It shuts down the floodgates.
Ah.
It downregulates its insulin receptors on the cell surface. It intentionally becomes energy deaf to the insulin signal. It is basically throwing up a blast door to stop the influx of glucose because it physically cannot process one more electron without destroying its own mitochondria.
Wow, that completely flips the script. The cell isn't broken, it's actively barricading the doors to survive the flood. But here's the problem with standard medical advice then. If my cells are shutting down the grid and throwing up blast doors to protect themselves from this toxic energy, why is the standard approach to prescribe medications that force the pancreas to pump out more insulin?
That's the million-dollar question.
Isn't that just battering a ram against the blast doors?
That is the absolute crucial tension in metabolic medicine right now. By forcing more insulin into the system, you might temporarily clear the sugar out of the bloodstream, which, makes your lab results look better on paper.
Right, the doctor is happy.
But you are often forcing that energy back into a cell that is already suffocating on toxic exhaust. You haven't fixed the mitochondrial backlog at all. You've just relocated the problem.
Which means we need a way to clear the backlog and repair the grid without using the glucose pathway.
Exactly.
And that brings us to the first massive biological lever in our sources, the metabolic switch of fasting and ketosis.
Biologically, when you enter a fasting state, say an overnight fast of 16 hours, or when you significantly restrict carbohydrates, you stop filling the reservoir. The water level drops.
So the flood ends.
Yes. Your body responds by burning through its stored glucose, which is called glycogen. Once that glycogen is depleted, your liver has to find a new fuel source. So it begins breaking down stored body fat into molecules called ketones, specifically one called beta-hydroxybutyrate, or BHB.
So we switch the entire city's power source from the flooded hydroelectric dam to, say, a wind farm. But what is actually happening to those damaged, leaking turbines at the quantum level when we make this switch?
This is where it gets amazing. A fascinating biological cleanup program initiates, known as quantum mitophagy. When the cell is deprived of incoming glucose, the nutrient-sensing pathways inside the cell, specifically one called mTOR, they shut down.
Okay, mTOR shuts down.
And this signals the cell that it is in a fasted, low-energy state. The cell immediately looks inward and says, okay, we have limited resources, we need to optimize our machinery.
It's doing an audit of the power grid.
An aggressive audit. The cell identifies the mitochondria that are damaged and leaking electrons. It tags them with a protein called ubiquitin. Then, an organelle called a lysosome, which is essentially a cellular recycling center, engulfs the damaged mitochondria.
It eats it.
It breaks it down into its base amino acids and lipids and uses those recycled parts to build a brand new, highly efficient mitochondria.
That is unbelievable. By just stepping away from the constant influx of food, your cells literally digest their own broken parts and build fresh engines.
It is the ultimate biological reset. You are physically repairing the cellular energy grid. And once the new grid is online, you start feeding it ketones instead of glucose. And the literature on ketogenic metabolism highlights that ketones are a significantly cleaner burning fuel.
Cleaner in what sense, though? I mean, an electron is an electron, right?
It's about how the electrons actually enter the chain. Ketones bypass the typical glycolysis pathway. They enter the mitochondrial energy cycle in a way that generates far more ATP per molecule of oxygen consumed.
Oh.
But more importantly, when ketones pass their electrons down the ETC, there is a drastically lower rate of electron slippage. You get all the power, the turbines spin beautifully, but you produce a fraction of that toxic RON-S exhaust.
So if you are staring at a pre-diabetes diagnosis, integrating fasting or periods of ketosis isn't just about losing weight. Like, it's a targeted mission to give your overwhelmed, barricaded cells a vacation.
That's a great way to put it.
You stop flooding the dam, you let the water recede, you trigger that lysosome recycling crew to repair the broken turbines, and you fuel the new engines with clean-burning wind power. And once the oxidative stress clears, the cell feels safe again. It opens the blast doors. It becomes sensitive to the insulin signal once more.
The biological logic is incredibly sound there. However, the sources are very clear that implementing a strict, permanent ketogenic diet presents major long-term structural problems for the rest of the body.
Yeah, and this is exactly where the narrative usually gets stuck. Anyone who has tried to live on zero carbohydrates knows the reality.
It's tough.
Aside from the fatigue and, the social isolation of never eating a carb at a restaurant, there is a massive biological flaw. If you starve your body of all carbohydrates, you are also starving the trillions of bacteria in your gut microbiome.
Right. The microbiome requires complex carbohydrates to survive. If you strip all of them away in the pursuit of perfectly flat blood sugar, those beneficial bacteria literally die off.
And that's bad news.
Very bad. When they die off, the lining of your gut degrades, which triggers systemic inflammation, which loops right back around to causing insulin resistance again.
That's a total catch-22.
Yeah.
You cut the carbs to save the mitochondria, but you kill the gut in the process.
Yeah.
So how do we thread this needle? How do we keep the blood sugar stable and the mitochondria clean while still feeding the microbiome?
That's the million-dollar question.
And the answer to that question is the entire focus of the massive meta-analysis published in the journal Frontiers in Nutrition. And it revolves around a structural anomaly called resistant starch.
Yes. To understand resistant starch, we have to look at the anatomy of human digestion. When you eat a standard carbohydrate, like a piece of white toast, a bowl of instant oats, or heavily processed pasta, the digestion actually begins in your mouth. Your saliva contains an enzyme called alpha amylase.
Alpha amylase is basically chemical scissors, right?
Exactly.
Yeah.
Starch is essentially just a long chain of glucose molecules linked together. Alpha amylase and other endogenous enzymes in your small intestine act like chemical scissors that rapidly snip those links apart.
Right.
Within minutes, the starch is dismantled into individual glucose molecules, which absorb directly through the intestinal wall and flood your bloodstream. That is your massive spike.
But resistant starch, or RS, behaves completely differently. It literally lives up to its name.
It does. It physically or chemically resists those enzymatic scissors. The enzymes just bounce right off. Because it cannot be broken down into glucose, it bypasses the small intestine entirely.
So no sugar spike at all.
None. It does not enter the bloodstream. It travels intact all the way down into the large intestine, the colon.
So the Frontiers meta-analysis reviewed 36 randomized controlled trials involving nearly 1,000 people with type 2 diabetes and prediabetes. And it breaks resistant starch down into 5 distinct categories. We want to focus intensely on type 1 and type 2 today, because the data shows these are the absolute heavyweights for managing glucose.
They really are.
Let's look at the actual architecture here. What is RS1?
Resistant starch type 1 is physically inaccessible starch. You find this in intact, whole grains, whole seeds, and whole legumes. The mechanism here is purely anatomical.
Okay, how so?
Every plant cell has a rigid, fibrous cell wall. In RS1, the starch is locked deep inside that cell. Your digestive enzymes simply do not have the physical tools to break down the plant's structural fiber.
So RS1 is like a locked safe.
Yeah.
The glucose is inside, the enzyme scissors are outside, but they can't pick the lock. The safe just passes entirely through the small intestine.
Exactly.
But this highlights a terrifying reality about our modern food supply. If the resistance depends on the cell wall being intact, what happens when a factory mills a grain into flour?
Well, when you subject grains to industrial processing, milling, grinding, extruding, you apply immense mechanical force and heat. You physically obliterate the plant cell walls. You blow the doors off the safe.
Oh, wow. So an intact oat groat is RS1. But the exact moment you grind it into oat flour, it's just raw glucose waiting to spike your blood.
Yeah, the chemical composition hasn't changed at all, but the structural integrity is totally gone. Once the cell wall is shattered, the alpha amylase has direct access to the starch. It snips it apart immediately.
That's crazy.
This is why highly processed foods are just devastating to the metabolic grid. The physical structure dictates the biological response.
Okay, that is RS1, the physically locked safe.
Right.
What about RS2? Because the data in the meta-analysis shows RS2 doing some incredibly heavy lifting for fasting insulin.
Yeah, resistant starch type 2 is native granular starch. You find this in raw high amylose maize, unripe green bananas, and raw potatoes. And the resistance here is fundamentally different from RS1. There is no protective cell wall holding the enzymes back.
Wait, if there is no cell wall, why can't the enzyme scissors just cut it?
It comes down to the molecular packing, specifically the ratio of amylose to amylopectin. Most starches have a lot of amylopectin, which is highly branched and super easy for enzymes to grab.
Like branches on a tree.
Right. But RS2 is incredibly high in amylose, which is a straight linear chain. Because it is linear, the amylose chains pack together extremely tightly, forming this dense B-type crystalline structure.
A crystal. So we are eating microscopic crystals of starch.
Effectively, yes. The amylose molecules bind to each other so tightly via hydrogen bonds that they completely squeeze out all the water. This is the crucial mechanism. Your digestive enzymes require water to hydrolyze or break those chemical bonds.
Oh, I see where this is going.
Because the RS2 granule is essentially waterproof, the enzymes literally slide right off the surface. They have zero grip.
That makes total sense. You can't dismantle a brick wall if your tools rely on water, and the wall is totally waterproof.
Uh-huh.
So what did the clinical trials actually show when diabetic patients consumed these locked safes and waterproof crystals?
The clinical effectiveness was remarkable. Both RS1 and RS2 drastically blunted postprandial blood glucose. So the dangerous spikes immediately following a meal. Because the starch isn't turning into glucose, there is no flood at the dam.
Right.
But the chronic intake of RS2 showed something even more profound. Over periods of 4 to 12 weeks, regular consumption of RS2 significantly lowered fasting insulin levels and improved HOMA-IR scores.
Let's break down HOMA-IR, because that is a term listeners might see on their blood work, but doctors rarely explain the mechanics of it to you.
Good idea. HOMA-IR stands for Homeostatic Model Assessment for Insulin Resistance. It's a mathematical calculation that takes your fasting glucose and your fasting insulin and multiplies them together. It gives us a snapshot of how hard your pancreas has to work just to keep your baseline blood sugar normal. If your HOMA-IR is high, it means your cells have their blast doors shut and your pancreas is screaming, pumping out massive amounts of insulin just to maintain the status quo.
And the meta-analysis proves that RS2 lowers this score. It essentially acts as a metabolic dampener, calming the pancreas down and helping the cells feel safe enough to open their doors.
Exactly. The standard mean difference in the data was highly statistically significant. By shifting the diet away from processed vulnerable starches and toward dense structurally resistant starches, you are fundamentally removing the oxidative stress from the mitochondria while still providing physical volume in the diet.
So the practical takeaway here isn't just eat fewer carbs. It is a radical shift toward examining the physical architecture of your food. You have to ask yourself, can I literally see the intact plant structure in what I'm about to eat?
Yes, that visual check is perfect.
Are you eating whole, intact lentils? Or are you eating highly processed lentil pasta? Are you eating whole rolled oats or instant oat dust? Preserving the structural matrix of the food is your first line of defense.
But the journey of the resistant starch doesn't end in the small intestine. The fact that it survives is really only the setup for the most profound biological reaction in our bodies.
Exactly. The locked safe and the waterproof crystal arrive completely intact in the large intestine. And this transitions us to our third level of analysis, the gut-incretin connection, where biology literally meets anatomy. Because the starch doesn't just sit in the colon doing nothing, does it?
No, it enters one of the most densely populated, highly active ecosystems on the planet. The human large intestine is this dark, anaerobic environment housing trillions of bacteria, and these bacteria are ravenous.
They're waiting for food.
They are. When the resistant starch arrives, specific strains of beneficial bacteria, most notably Faecalibacterium prausnitzii, Roseburia, and Bifidobacteria, they recognize it. While your human enzymes couldn't break the structure, these bacteria possess highly specialized enzymes that can.
They crack the safe.
They do. They latch onto the resistant starch and begin a process of fermentation. They are literally eating the starch for their own survival. But it's what they excrete as a byproduct of this fermentation that changes human health. They produce short-chain fatty acids, or SCFAs.
The sources single out three major SCFAs: acetate, propionate, and butyrate. I want to drill all the way down into butyrate because the literature reads like a love letter to this specific molecule. What makes butyrate so magical at a cellular level?
Structurally, butyrate is a very small fatty acid chain, only 4 carbon atoms long, but its primary role is serving as the absolute preferred fuel source for your colonocytes, the cells that physically line the wall of your colon. They don't want glucose, they want butyrate.
Wait, my gut cells eat the fat excreted by the bacteria that ate the starch.
It is a perfect symbiotic loop. When the colonocytes absorb the butyrate, they use that energy to fortify the intestinal barrier. Your gut lining is only one single cell thick, and these cells are held together by proteins called tight junctions.
Like the mortar between the bricks.
Precisely. In a state of poor metabolic health, those tight junctions degrade. The mortar crumbles. This causes intestinal permeability, commonly known as leaky gut.
Which sounds awful.
It is. Microscopic particles of undigested food and bacterial endotoxins, specifically a nasty one called lipopolysaccharide, or LPS, they leak out of the colon and directly into your bloodstream.
And when LPS hits the bloodstream, your immune system just loses its mind.
It triggers a massive systemic inflammatory response. The immune system floods the body with cytokines. And here's the direct link to our main topic today. Systemic inflammation aggressively drives insulin resistance.
How does it do that?
The inflammatory cytokines travel to your muscle and liver cells and physically interfere with the insulin signaling pathways. Even if the cell wants to open the blast doors, the inflammation jams the lock.
Wow. So let me trace this back. By feeding the bacteria-resistant starch, they produce the butyrate. The butyrate repairs the mortar in the brick wall, the LPS stops leaking into the blood, the immune system calms down, and the locks on the cellular blast doors are finally cleared of the inflammatory gunk.
That is the direct mechanical pathway, yes. But butyrate does something even more advanced inside the human body. Once it is absorbed, it travels to cells and interacts with a protein complex called KEAP1. Normally, KEAP1 acts as an anchor, holding back a master transcription factor called NRF2.
We talked about NRF2 earlier in the show as the master regulator of antioxidants. How does butyrate trigger it?
Butyrate literally causes the KEAP1 anchor to release. NRF2 is then free to travel directly into the nucleus of the cell, where it binds to the DNA at specific sites called antioxidant response elements. It turns on the genes that manufacture your body's innate internal antioxidants like glutathione.
So the butyrate from the gut travels to the cells, hacks the DNA, and orders the factory to produce a massive wave of antioxidants to go clean up the toxic raw and test exhaust we talked about in the mitochondria.
It is a highly coordinated biological defense network, and it all starts with the structure of the starch you put in your mouth.
That alone is mind-blowing. But the sources point to another mechanism in the gut that is totally dominating the medical news right now. There is a study from the LSU Ag Center on rodents that tracks how resistant starch fermentation impacts GLP-1.
Ah, yes.
If you turn on a television right now, you will see a commercial for a GLP-1 drug. Ozempic, Wegovy, Mounjaro. These are GLP-1 receptor agonists. How on earth does a dietary starch connect to the hottest weight loss and diabetes drugs on the market?
To understand this, we have to look at the anatomy of the intestinal wall again. Interspersed among those colonocytes we just talked about are highly specialized endocrine cells called L cells. These L cells have receptors on their surface, specifically FFAR2 and FFAR3, which stand for free fatty acid receptors.
Let me guess what binds to those receptors.
The short-chain fatty acids. When the bacteria ferment the resistant starch and produce massive amounts of propionate and butyrate, those SCFAs bind directly to the FFAR receptors on the L cells.
And what happens then?
This binding triggers an intracellular cascade involving calcium signaling, which culminates in the upregulation of the proglucagon gene. The cell manufactures and then secretes a hormone, endogenous GLP-1.
Glucagon-like peptide 1. What exactly does this hormone do once it hits the bloodstream?
It is an incretin hormone, meaning it acts as a massive metabolic coordinator. It travels to the pancreas and tells the beta cells to secrete insulin, but only in a highly controlled glucose-dependent manner so it won't crash your blood sugar.
That's smart.
Very. Simultaneously, it travels to the stomach and slows down gastric emptying, meaning food digests much slower. And finally, it crosses into the brain, specifically the hypothalamus, and flips the satiety switch. It literally tells your brain, we are full, stop eating.
Okay, I have to challenge this, though. I understand the biological mechanism. But pharmaceutical companies are injecting massive, synthetic, heavily engineered doses of GLP-1 agonists into people to achieve these effects. You are telling me that eating a cold potato or an unripe banana is going to do the exact same thing. How can a dietary starch compete with a billion-dollar pharmaceutical injection?
It's an essential distinction to make, and it all comes down to pharmacokinetics, the timing, the half-life, and the delivery of the molecule. When a patient takes a pharmaceutical GLP-1 injection, they are getting a massive supraphysiological dose all at once.
Right, a huge spike.
And those synthetic molecules are engineered to resist degradation, meaning they linger in the blood for days. This overwhelming spike is exactly why patients often experience such severe side effects: nausea, vomiting, gastric
The
paralysis.
system is just being completely flooded.
Contrast that with endogenous GLP-1 produced by resistant starch fermentation. Naturally produced GLP-1 has a very short half-life in the blood. It is rapidly degraded by an enzyme called DPP-4. If it were released all at once, it would be gone in minutes. But remember how fermentation works.
Right, the bacteria have to slowly chew through the waterproof crystals in the locked safes.
Exactly. The fermentation process is incredibly slow. It takes hours and hours as the starch moves through the winding anatomy of the colon. Because the bacteria are constantly chewing and producing SCFAs, those L cells are constantly being stimulated.
That's like a time-release capsule.
It is a continuous, natural, sub-pharmaceutical drip of GLP-1 occurring over the entire day and night. You don't get the violent spikes and nausea of an injection. Instead, you elevate your baseline physiological level of GLP-1.
And what did the LSU study show about that baseline?
This study demonstrated that this sustained, steady presence of the hormone is profoundly effective at improving long-term glycemic homeostasis. Over time, it actually promotes the repair and proliferation of the pancreatic beta cells themselves.
This is a massive paradigm shift. By feeding the specific strains of bacteria deep in your colon, you are literally cultivating your own internal pharmaceutical factory. You are paying the bacterial workers in resistant starch, and they are manufacturing and pumping out steady streams of blood sugar stabilizing, appetite suppressing hormones 24 hours a day.
It really showcases the profound elegance of human anatomy when it is supplied with the correct structural fuels. The SCFAs seal the gut, they quench the systemic oxidative fire via NRF2, and they deploy the incretin hormones that fine-tune the insulin response.
All right, we've repaired the quantum grid, we've locked the structural safes, and we've fired up the gut fermentation factory. Let's move to the fourth level of our deep dive, the psychological intersection.
This is where it gets really applicable to daily life.
Because if you're living with prediabetes, the lab numbers are kind of abstract. What isn't abstract is how you feel. We need to talk about the brain. If we fix the gut and stabilize the physical spikes, why do so many people with metabolic issues still struggle with severe brain fog, anxiety, and mood swings?
Because the brain is the most energy-demanding organ in the human body. It represents only about 2% of your body weight, but it consumes 20% of your energy. When the energy grid fluctuates, the brain is the first to panic.
Makes sense.
The sources pull from peer-reviewed neurological studies, including a retrospective analysis from the journal PeerJ, that explicitly link high glycemic variability to severe cognitive and emotional degradation.
Let's define glycemic variability. It's the roller coaster, right? It's not just about having high blood sugar. It's about the violent spikes, followed by the crashing lows.
The volatility is exactly what causes the damage. The PeerJ study evaluated middle-aged and elderly patients with high glycemic variability and found striking correlations with tension, anger, depression, and rapid cognitive decline.
Did they test their cognition?
Yes, these patients scored significantly lower on the Montreal Cognitive Assessment, or MoCA, which tests memory, executive function, and attention.
Walk me through the anatomy of that damage. How does a sugar spike in the blood translate to brain fog and anger in the mind?
It begins with the microvasculature. The blood vessels supplying your brain are incredibly fine and delicate. When a massive wave of glucose hits those vessels, it causes glycation. The sugar molecules literally bind to the proteins in the blood vessel walls, making them stiff and brittle.
And that damages the barrier, doesn't it?
Yes, it damages the blood-brain barrier.
The blood-brain barrier is supposed to be the ultimate security checkpoint, keeping toxins out of the brain tissue.
But the extreme glucose fluctuations compromise it. This leads to massive oxidative stress, more of those toxic RONs we discussed, erupting directly inside the brain tissue. The brain's immune cells, called microglia, activate and trigger neuroinflammation. And when the brain is physically inflamed, the neurons struggle to fire correctly. The communication grid starts short-circuiting.
Which brings us to the neurochemistry of mood. The sources spent a lot of time mapping out two specific neurotransmitters, glutamate, and GABA. How do these two chemicals dictate our daily emotional state?
You can think of glutamate and GABA as opposing forces. Glutamate is the brain's primary excitatory neurotransmitter. It is the accelerator. When glutamate binds to a neuron, it makes that neuron more likely to fire. We need glutamate for learning, memory, and focus, but you cannot have a brain that is constantly accelerating.
Because that leads to panic, right?
Precisely. That is where GABA comes in.
Hmm.
Gamma-aminobutyric acid is the primary inhibitory neurotransmitter. It is the brake pedal. When GABA binds to a neuron, it hyperpolarizes the cell, making it less likely to fire. It calms the nervous system, reduces anxiety, and prepares the brain for sleep. A healthy brain requires a flawless, constant balancing act between these two chemicals.
Let's use an orchestra analogy here. Glutamate is the brass section playing a triumphant, aggressive march. GABA is the conductor raising his hands, bringing the tempo down, and signaling the string section to play a soothing melody so the music doesn't just evolve into pure, chaotic noise.
That is a perfect analogy. But here is the tragedy of a modern, highly processed diet. The neuroinflammation and the violent blood sugar spikes impair the enzymes responsible for regulating this balance. The brain gets stuck in a state the literature calls glutamate dominance.
So the brass section is playing as loud as they can, and the conductor has left the building.
The accelerator is jammed to the floor. The brain is flooded with excitatory signals. Psychologically, this manifests as chronic, low-grade anxiety. You feel wired but tired. You suffer from racing thoughts, irritability, and an inability to focus on a single task. Your brain is essentially redlining its engine all day long.
Okay, this brings us back to the incredible synergy of our sources.
Mhm.
How does the metabolic switch we discussed in Section 1 shifting into a state of ketosis by utilizing fasting and resistant starch actually reach into the brain and fix this glutamate dominance?
Because ketones, specifically beta-hydroxybutyrate or BHB, cross the blood-brain barrier completely differently than glucose. They use a different doorway, specifically the monocarboxylate transporters. When the brain shifts from burning volatile glucose to burning clean BHB, the neurochemistry fundamentally alters.
How does BHB affect the orchestra?
It acts directly on the enzymes that manage the neurotransmitters. BHB upregulates the activity of an enzyme called glutamate decarboxylase, or GAD. The GAD enzyme has one specific job. It physically takes the excitatory glutamate molecule, removes a carbon atom, and converts it directly into calming GABA.
Wait, let me make sure I understand the magnitude of this. The ketone doesn't just block the anxiety chemical. It physically captures the chemical causing the anxiety, dismantles it, and transforms it into the exact chemical that causes calm.
It transforms the stressor into the solution. Ketosis actively drains the excitatory pool and fills the inhibitory pool. It brings the conductor back to the podium. Furthermore, because the BHB is burning so cleanly in the mitochondrial turbines, the neuroinflammation subsides. The blood-brain barrier heals.
That's incredible.
The daily psychological result is a profound sense of emotional resilience, reduced panic, and crystal clear mental focus. You regain control of the accelerator and the brake.
And the ketones aren't working alone to rebuild the brain, are they? The sources introduce another molecule that honestly sounds like science fiction. BDNF.
Brain-derived neurotrophic factor. If ketones are the clean fuel, BDNF is the architectural blueprint for a better brain. BDNF is a protein that acts like Miracle-Gro for your neurons. It stimulates neurogenesis, the actual growth of new neural pathways, and protects the synapses you already have.
And how do we trigger the body to produce this Miracle-Gro?
Ketones inherently stimulate BDNF production. But the most powerful lever you can pull is physical exercise. And this connects beautifully to glucose management. When you engage in exercise, your muscles require immense amounts of energy. Usually they need insulin to open the doors.
Right.
But exercise activates a metabolic pathway called AMPK.
We briefly mentioned AMPK in the outline. What is the actual mechanism here?
AMPK is an energy sensor. When it detects that the muscle is contracting and burning ATP, it essentially kicks open a back door to the cell. It allows the muscle to pull glucose directly out of the bloodstream, completely independent of insulin.
So even if your cells have their blast doors shut and your insulin resistance is super high, exercise bypasses the lock entirely and vacuums the sugar out of your blood.
Yes, and as the AMPK pathway clears the glucose, the physical exertion simultaneously triggers a massive release of BDNF into the brain. You clear the metabolic backlog and fertilize the neural networks in one single action.
It's all woven together so perfectly. The muscle contractions, the blood sugar clearance, the ketone fuel, the neurogenesis. But there is one final anatomical piece to this puzzle. A factor so powerful it can completely override a perfect diet and a perfect exercise routine. We have to talk about stress.
This is the invisible variable that derails so many people, especially those who are newly diagnosed. Getting told you have a chronic metabolic condition is deeply stressful. You worry about your future, your diet, your vision.
Yeah, the anxiety is real.
And that psychological worry instantly becomes a biological reality through the hypothalamic pituitary adrenal axis, or the HPA axis.
Trace this signal for me. How does feeling anxious about, say, an email from my boss or worrying about a medical bill actually raise my blood sugar?
Let's follow the anatomy. When your brain, specifically the amygdala, perceives a threat, whether it's a physical predator or psychological stress, it sends a distress signal to the hypothalamus. The hypothalamus acts as a command center and releases a hormone called CRH.
Okay.
This travels a fraction of an inch to the pituitary gland, which then secretes ACTH into the bloodstream.
And where does the ACTH go?
It travels down to your adrenal glands, which sit right on top of your kidneys. The ACTH signals the adrenal cortex to flood your system with cortisol, which is the body's primary stress hormone.
And what is the physiological mandate of cortisol?
Survival. From an evolutionary perspective, cortisol prepares you to fight or flee. To do that, your skeletal muscles require instant explosive energy. But the brain doesn't know you're just sitting in traffic. It thinks you are running from a tiger.
Right.
So cortisol travels to your liver and binds to glucocorticoid receptors.
This is where the sabotage happens.
It alters the DNA transcription in the liver cells to massively upregulate an enzyme called PEPCK. This enzyme drives a process called gluconeogenesis. Your liver literally starts dismantling proteins and amino acids, reassembling them into pure glucose molecules, and forcefully dumping them directly into your bloodstream.
Let me pause here because this is the most frustrating realization for anyone trying to manage their metabolism.
Yeah.
You could be perfectly adherent to your diet. You could be fasting. You could be eating zero carbohydrates. But if you are trapped in a state of chronic psychological stress, your own liver will continually manufacture sugar and dump it into your blood, driving up your insulin resistance from the inside.
You are basically fighting a war against your own liver. And it gets worse. Cortisol also directly binds to your muscle and fat cells and makes them more insulin resistant.
So it's a double whammy.
It is. The body is intentionally keeping the sugar circulating in the blood so it's available for the emergency. If the HPA axis is constantly firing, dietary interventions will fail.
So we need an anatomical tool to hack this system. If the HPA axis is the panic button, where is the biological off switch?
The off switch is the vagus nerve. The vagus nerve is the 10th cranial nerve, and it is the superhighway of the parasympathetic nervous system, the rest and digest protocol. It wanders from your brainstem down through your neck and directly interfaces with your heart, your lungs, and your digestive tract.
The sources outline a surprisingly accessible way to manually stimulate this nerve: diaphragmatic breathing. How does breathing change the liver's glucose production?
It's pure mechanical signaling. When you take shallow, rapid breaths from your chest, you signal panic. But when you intentionally take slow, deep breaths that expand your diaphragm, your belly, the physical stretching of the diaphragm activates the stress receptors attached to the vagus nerve.
You're physically tugging on the nerve.
And when stimulated, the vagus nerve releases a neurotransmitter called acetylcholine. This chemical signal races up the superhighway directly to the hypothalamus. The message is simple and profound. The tiger is gone. We are safe.
And when the command center receives that safety signal,
It immediately halts the production of CRH. The domino effect stops. The adrenal glands stop pumping cortisol. As cortisol levels plummet, the liver receives the signal to shut down gluconeogenesis.
that's amazing.
The internal glucose dump just ceases. Furthermore, the acetylcholine released by the vagus nerve binds to receptors on your immune cells, shutting down the production of inflammatory cytokines.
You literally breathe the inflammation away.
You halt the stress-driven glucose spike, you quench the systemic inflammation, and you drastically improve your heart rate variability, or HRV, which is the ultimate metric of a resilient, adaptable nervous system.
Okay, we have excavated this topic all the way down to the bedrock. We have looked at the quantum electron leakage in the mitochondria, the structural architecture of locked safes and waterproof starch crystals, the biological fermentation factories in the colon, the orchestra of neurochemistry in the brain, and the anatomical wiring of the stress response.
We've covered a lot of ground.
Now, we must fulfill our promise to you, the listener. We need to synthesize this into a practical daily roadmap tailored for someone staring down a diagnosis of prediabetes or type 2 diabetes.
The foundation of this roadmap is synergy. It is about understanding how these mechanisms support each other. If you only apply one lever like a strict, permanent ketogenic diet, you clear the mitochondrial backlog, but you starve the microbiome, leading to leaky gut.
Right.
If you only eat complex starches, you feed the gut, but you might still be flooding an already overwhelmed quantum grid.
So the solution is an engineered combination. The metabolic reset of ketosis merged with the targeted structural feeding of resistant starch.
Exactly. By adopting a low glycemic, modified ketogenic approach, you restrict the rapidly digestible carbohydrates. You starve the body of excess volatile glucose. This lowers the insulin demand, allowing the cellular blast doors to open.
And that triggers the reset.
It triggers the lysosomal mitophagy to recycle the broken turbines, and it fuels the brain with clean-burning BHB to convert the anxious glutamate into calming GABA.
But into that low glycemic landscape, we strategically deploy the resistant starches.
We deploy the RS1 and RS2 because they bypass the small intestine entirely. They do not spike the blood sugar. They do not contribute to the mitochondrial bottleneck. They arrive perfectly intact in the colon, where they keep the bacterial ecosystem thriving.
The SCFAs from the fermentation beautifully complement the ketones. The ketones fix the brain and the mitochondria, while the butyrate from the starch heals the leaky gut, drops the systemic inflammation, and provides that endogenous time-release drip of GLP-1 to repair the pancreatic beta cells. It is a masterpiece of biological engineering.
It really addresses the pathology comprehensively, from the single cell to the entire organism.
Let's get incredibly practical. If you are listening to this right now, how do you actually eat this way? I'm going to list 3 specific, highly accessible, resistant starch food sources that you can integrate into a low glycemic routine today. Number 1, cooked and completely cooled potatoes.
This introduces a fascinating third type of resistant starch, RS3, which we haven't discussed yet. RS3 is created through a process called retrogradation.
How does that work?
When you boil or bake a potato, the heat and water gelatinize the starch, making it highly digestible. But if you place that potato in the refrigerator and let it cool completely for 24 hours, the starch molecules physically realign. They form tight crystalline bonds that resist digestion.
So a hot-baked potato is a massive blood sugar spike, but a cold potato salad, perhaps mixed with some extra virgin olive oil for healthy fats, is a locked safe that feeds the microbiome.
Yes, the cooling process fundamentally alters the architecture of the food.
Food source number 2, green unripe bananas or green banana flour.
This is your raw, dense RS2. We all know what a yellow, spotty banana is. It's basically nature's candy. The starch has fully converted to simple sugars. But a firm green banana is packed with native granular starch, those waterproof crystals we discussed.
How do you eat it, though?
You can slice a green banana into a low-sugar smoothie or bake with green plantain flour. It acts as a powerful metabolic dampener, steadily lowering that fasting HMA-IR score.
And food source number 3, uncooked oats.
This utilizes the physical barrier of RS1. If you cook oats into a hot bowl of oatmeal, the heat and water explode the plant cell walls. The safe is blown wide open, and you get a rapid glucose spike.
And if they're raw?
You take raw, rolled oats and soak them overnight in almond milk or blend them raw into a protein shake. The cellular matrix remains intact. The enzymes bounce off, and the starch reaches the bacterial factory in the colon.
Okay, let's assemble the daily routine. The accessible roadmap to put the quantum, biological, anatomical, and psychological lenses into action.
The protocol begins before you even wake up. You utilize a daily overnight fasting window, typically 14 to 16 hours, where you consume no calories.
So if you finish dinner at 7 p.m., you don't eat again until 9 or 11 a.m.
Correct. This prolonged absence of incoming energy drains the liver's glycogen reservoir. It shuts down the mTOR pathway, signaling the lysosomal recycling crew to initiate quantum mitophagy. You clean up the damaged mitochondria and drift into a mild state of ketosis, providing clean BHB fuel to the brain.
When you do break the fast, you avoid the massive carbohydrate spike. You prioritize healthy fats, proteins, and fibrous greens. An omelet with spinach and avocado, for example.
This sustains the ketone production and prevents a violent glycemic roller coaster. You maintain the conversion of excitatory glutamate into calming GABA. Your brain remains anchored, focused, and emotionally resilient throughout the most demanding parts of your day.
Then, perhaps at lunch or dinner, you deploy the resistant starch. You incorporate a serving of the cooled potatoes, the green banana flour, or some whole legumes.
You deliver the intact structural matrix to the colon. The bacterial fermentation factory powers up. The butyrate seals the gut lining, activates the NRF2 antioxidant defense, and the L cells begin secreting that slow, steady, subpharmaceutical drip of endogenous GLT-1, fine-tuning your insulin sensitivity while you sleep.
And finally, layered throughout the entire day, you use your anatomy. When the emails pile up, when the traffic stops, when the panic of the diagnosis sets in, you pause. You execute 5 minutes of deep, diaphragmatic breathing.
You manually stretch the vagus nerve, you flood the brain with acetylcholine, you silence the command center, shut down the HPA axis, and stop the cortisol from forcing your liver to sabotage your blood sugar.
You are simultaneously acting as the quantum mechanic, the gut gardener, the neurological conductor, and the anatomical engineer. It is total, comprehensive metabolic management. We've thrown an immense amount of science at you today, but I hope what emerges from these sources is a profound sense of awe at how interconnected your body truly is. Managing your blood sugar is not a punitive diet of restriction. It is a conscious, deeply empowering tuning of your biological instruments.
And as we close, I really want to leave you with one final paradigm shift to ponder. At the beginning of this deep dive, we talked about discarding the idea that your cells are malfunctioning.
Right.
When you look at the quantum reality of insulin resistance, the cell intentionally downregulating its receptors, closing the blast doors to protect the mitochondria from the toxic exhaust of caloric overload, you realize something profound.
What is that?
Insulin resistance is not a disease. It is a brilliant, desperate evolutionary adaptation. Your cells evolved over millions of years to survive famine, not to process a ceaseless avalanche of processed industrial calories.
That makes total sense.
When faced with this unprecedented toxic energy overload, your body isn't failing you. It is executing a flawless defense protocol. It is throwing up a shield to keep you alive. When you change the architecture of your fuel by utilizing clean fasting, ketones, and resistant structural starches, you aren't fighting a disease. You are simply giving your miraculous cells the safety signal they need to finally lower their shields.
You aren't broken.
Yeah.
Your shields are just up. We want to enthusiastically thank you for joining us on this incredibly deep dive today. You now have the mechanistic understanding of exactly how your metabolism operates, and you have the tools to tune it. We encourage you to take just one small step for the roadmap today. Put those potatoes in the fridge, extend your fast by an hour, or just take 5 deep, vagal breaths. You are in control of the grid. Until next time, keep diving deep.